Welcome to the fourth video of the Coral Conservation Series with the Texas State Aquarium. In our last video, you joined me live in Hawaii during our winter experiments where we learned more about the experiments that we were doing. During this video, you'll learn more about what we did. We collected samples when we were there and how we're processing those samples, and also how we're ramping up for our another expedition to Hawaii where you'll be joining me again live there. Our last trip in Hawaii was a very successful trip. We were able to collect different species of corals. We looked at 2 species of corals and we conducted our experiments. We did 2 back-to-back experiments. Both of them were 30 days long, which is very, very time intensive, of course. And during those 30 days, we do 2 overnight experiments at the same time, where we look at how corals are growing and what the health is of them over a 30-hour period. When we were there, we were really successful. We were able to collect all the corals we wanted, and we also took samples of the corals so we could look at what's going on inside of the coral and how well is that symbiosis or that relationship between the coral as an animal and the algae or the symbiont inside of the coral. So when we were there, some of the major accomplishments that we got the data that we needed to get, which is really important. And we also saw that all of our corals were really healthy and they continue to grow even though we changed the chemistry of the water. So we're very successful. We were able to collect that data and also train some students when we were out there at the same time. So one of the things that we didn't accomplish that we would have liked to accomplish when we were there was to get some more footage of what the coral reefs looked like prior to this summer, and we're gonna plan on doing some surveys when we're back out there in a few weeks so we can get an idea of what the current health of the corals on the reefs are, and we can see how that might be changing. Since it is the summertime, it is going to get warmer, and we are a little bit concerned that we might have some too much warming going on. We might see some bleaching of the corals. So we want to get an idea of what that current health status is of the corals, so we'll be doing that this summer. We weren't able to do that during the winter time since we were so busy getting these experiments up and running. So when we came back during the winter time, so we came back in February, we wanted to get an idea of how the corals might have been responding to the treatments that we exposed them to when we changed the chemistry of the water. So we wanted to take fragments from each of the different corals that we worked with. So before and after the experiment, we took just a small sample from each of the corals, the 2 different species that we were looking at, and we brought those back. They're frozen and we processed those. We're currently processing those to see what the corals themselves look like and what the algae inside of them look like and how much zooxanthellae or the symbiont are inside the coral and how much chlorophyll is inside of those symbionts as well. Once we grab it out of the freezer, what we'll first do is remove the tissue of the coral from the skeleton. And we do that by airbrushing or using some kind of force pressure of water to remove that tissue, and we collect it inside of a plastic bag. So once we collect all of the coral tissue inside of the plastic bag, we are then going to place that into a larger tube so we can mix it up. And the reason why we're mixing this up is because we want to be able to separate the algae from the coral. And that will allow us to look at the coral by itself and the algae by itself. So we can get a metric or a measurement of how healthy the coral is and how healthy the algae are inside of the coral. After we're done mixing it up, then we're going to separate it and using a centrifuge, which is just a really fast way to spin everything down. And what you'll see is at the bottom of the centrifuge, or bottom of the tube, is the symbiont or the algae. And then in the rest of the sample is the coral. So we're going to separate those. We're going to take a sample of the coral, so we have that for later for our analyses, and then we're going to then dump out all of that coral, and we're then going to just have the symbiont by itself. Once we have that, we're going to resuspend that pellet or resuspend those algae in some nice clean water, and then we'll take subsamples of those algae so that we can look at them under the microscope, count how many they are, and then we're also going to quantify how much chlorophyll is inside of those symbionts as well. Now that we have a sample of the zooxanthellae, what we need to do is put it into a cell counter or a hemocytometer so we can look at how many algae or how many individual cells of the zooxanthellae do we have. So you can see here that we will have a ton of symbions in this sample, and what we are going to do is go through and count every single one of those cells, or every single one of these green or yellowish color circles that you see here on this microscope slide. So this could take a good amount of time, but we really want to know is how much zooxanthellae does each of these corals have? And that gives us an idea of how healthy that coral is based on how many zooxanthellae it has. So after we're done doing all the processing with the coral tissue, what we do with the skeleton is we want to see how that coral has been growing its skeleton over time. The way that we do that is we place the coral skeleton in a known volume of water, as you can see here, and we look at how much of it is displaced. And then we measure the mass of the mass of the coral skeleton, and then that gives us an idea of the overall density of the coral. So we look at the density of the skeleton of the coral for each coral that we collected, and we look at it across different treatments to see if the treatments are impacting how corals are growing their skeleton. So our next trip is taking off in just a few short weeks here, and what's really exciting about this trip is we're bringing students from Texas A&M Corpus Christi, and we're also bringing students from the University of Hawaii at Manoa. So we're repeating the same experiments that we did in the wintertime, but now in the summertime. And since it is the summertime, the water's going to be warmer, so we might see some bleaching with the corals, and we want to make sure that we pay particular concern or attention to that to make sure that if there is bleaching that we're documenting that with the corals. But what's really exciting about this summer is that the students actually get to participate in this research and come up with some of their own projects to answer some of these questions that we're seeing with how the chemistry of the water might be influencing the coral's growth. And another thing on top of that is they have the opportunity to stay at a state-of-the-art marine institute, and not a lot of people have had this opportunity to stay there and work alongside researchers. So while we're there, we're going to be immersing them not only in the research, but working on their professional development skills and exposing them to other types of research that is going on at the same time. So the biology of the corals, the physiology, but also the engineering and the chemistry side. And these students that we're bringing from Texas and from Hawaii, they have different backgrounds in what they're studying. Some of them are studying biology, some of them are studying environmental science, some of them are studying engineering. So we're bringing all these different students from different backgrounds and disciplines together, putting them on the island along with myself, and we're going to be working for the next 4 weeks to really investigate what's going on with our coral reefs and hopefully be able to come to a consensus and come up with some solutions of how we can make sure that these coral reefs are there for future generations.
Hi, my name is Ignacio, and I am an undergraduate researcher in the Bahr Marine Ecology Lab. My favorite part of being here in the laboratory is collaborating with other researchers and growing as a researcher and learning more about coral reefs. I am most looking forward to is the amazing weather, the beautiful clear waters, and of course being immersed in all the corals that are within the Hawaii region.
Hi, my name is Eleanor, and I'm an undergraduate researcher in the Bahr Marine Ecology Lab. My favorite part about being in the Bahr Marine Ecology Lab is gaining practical research experience so I can go to graduate school. What I'm looking forward to in Hawaii is seeing an actual coral reef. I've never seen one in person, and I'm really excited to see one.
So I hope that you'll join me and the Texas State Aquarium this summer as we go live in Hawaii once again to learn more about the research that we're doing and how the students are involved in those research. So stay tuned, and we'll be seeing you there.